selection. And this is problematic because as populations become increasingly small and isolated, opportunities for gene flow may decrease, which may lead to increased inbreeding and decreased genetic diversity in the fragment populations. A number of different studies have shown that gene flow is really important for maintaining levels of genetic diversity in populations, as well as governing levels of genetic differentiation among populations. This idea of genetic rescue, where even a single immigrant can greatly improve population numbers and fitness, have been demonstrated in a number of different species, including the prairie chicken and mountain lion. However, longitudinal studies of a single genetic diversity in a single population through time remain rare, and there aren't that many cases where there have been documented genetic and fitness consequences of decreased immigration into a single population through time in nature. And so the question that I wanted to ask was, okay, given a demographically stable study population that is relatively large, if the regional surrounding populations decline in size, does this lead to decreased immigration into our focal population, and does that then lead to decreased genetic diversity? So the system that I use is the Florida Scroce, Aphelectoma coriolescens. It's a cooperative breeder. It's non-migratory and highly philopatric, which is nice because individuals that are born in our study tract tend to stay there, and so we can follow the same individuals throughout their lifespan. They're socially and mostly genetically monogamous. We've just discovered a really, really low extra-pair paternity rate. But for the most part, the pedigrees that we observe in the field are accurate, and so we can construct pretty accurate relationships for all of our individuals. You can't really see this. Well, the Scroce is restricted to this really beautiful and unique xeric habitat found in central Florida along the Lake Wales Ridge. And this habitat has been decreasing significantly due to humans constructing cattle ranches and citrus groves and strip malls. And so because of this, the Florida Scroce is now listed as a federally threatened species. Its population numbers have declined significantly through time. So there's been a 97% decline in the past century and more than a 50% decline in just the past 20 years. The reason I'm studying this species is that there's been a single population that's been monitored intensively at Archbold Biological Station in Florida since 1969. And I should say now that none of the work I do would be possible without the dedication of all the staff and field crew that have done a lot of field work at Archbold throughout the years. And so there's a lot of field work that goes on at Archbold. So every single month our entire population is census, so we can get detailed lifespans for every individual. Every nest of every family group is found and monitored, and so we can actually measure annual and lifetime reproductive success. All new immigrants and nestlings recruited into our population are banded and measured, and from 1988 through 1999 and from 1988 to 1995 and from 1999 to the present, we've collected blood samples from every single individual in our population. All the territories are mapped every year, and we also have data on habitat composition, weather, food availability, and fire history. So what this means is that we have this really rich sample. So we have blood samples from more than 4,000 individuals with complete life histories and morphometric data on this 12-generation pedigree. And all we need, so this makes the Florida Scroce really uniquely suitable for evolutionary population genomic studies, as long as we have genomic data. So during my PhD, what I did was I developed genomic resources for the species. I used genotyping by sequencing to discover a panel of genome-wide SNPs. We've sequenced and assembled the genome assembly and also the transcriptomes of diverse tissues. And what I'm doing right now is building a really detailed linkage map and annotating our genome. Today, however, I'm only going to talk about our genotype data. So we used custom Illumina iSelect B chips to genotype 3,800 individuals at 12,000 SNPs. So here I'm showing you the number of individuals in our population through time, with breeders on top and nestlings on the bottom. So gray bars indicate the total number of individuals, and blue bars indicate the number of individuals that were genotyped each year. So as you can see, we have almost complete sampling. We've genotyped almost every single bird in our population in the past 10 years. And so the regional habitat around Archbold has remained really stable. Population numbers have remained stable. They've even increased slightly through time, mostly because of really good habitat management and conservation efforts. But the peripheral populations surrounding our study population have been declining drastically, and a lot of them have gone extinct over the past few years. And so the first thing we wanted to ask was whether or not we see a decrease in immigration rate into our population. Our study track was purposely expanded in the late 1980s, so I'm going to start all of my analyses in 1990. Since all of our individuals are banded, we can identify immigrants coming into our population each year. And indeed, we do find that the number of immigrants coming into our population each year is decreasing through time. There's a bit of wiggle, but the trend is significant. What's probably more important for this particular study is the total proportion of breeders in a given year that were born outside our study tract. And that has decreased pretty dramatically from about 45% in 1990 to below 25% in 2013. So to investigate the genetic consequences of this decreased immigration, we used about 7,800 autosomal SNPs in approximate linkage equilibrium to estimate genome-wide levels of heterozygosity and inbreeding coefficients for every individual in our population. We also calculated pairwise identity by descent values for every pair of individuals, and so this is basically a measure of how related these two individuals are. So the first thing we looked at was heterozygosity of the different breeders. So here I'm showing you mean observed heterozygosity for years in which we have at least 50 breeders that had genotype data, with immigrants shown in yellow and residents shown in blue. And you can see that immigrants are clearly less heterozygous than our residents. So this could be due to two things. One, it's possible that our immigrants are coming from smaller, more isolated, and presumably more inbred populations. Another is that it's possible that the birds that do disperse just tend to be more homozygous. But we need to do a lot more sampling to try to disentangle this too. Even though these immigrants are less heterozygous, they are important in contributing genetic diversity to our population. So here I'm showing you the mean pairwise ID, so how relatedness of the breeding pairs through time. So in green I'm showing you all pairwise, all possible male-female combinations for that given year. In yellow are observed immigrant pairs, so these are mated male-female pairs where at least one individual is an immigrant. And then in blue, I'm showing you the observed male-female pairs where both birds were born in our study tract, both birds were residents. And you can see that there's the immigrant pairs tend to be less related than average. And we see evidence of inbreeding in the resident-resident pairs. This is kind of hard to see, but this line is kind of increasing through time. And so if the parents in our population are becoming more related through time, then we would expect that the nestlings are becoming more inbred. And so we looked at this in two ways. First, we looked at the mean inbreeding coefficient of the birth cohort in relation to the proportion of breeders that are immigrants in that particular year, and we find that they're strongly negatively correlated. We also looked at the mean inbreeding coefficient of the birth cohort through time, and it is increasing slightly but significantly through time. The pattern gets a little bit more interesting if you classify the nestlings into those who had two resident parents versus nestlings that had at least one immigrant parent. So here, green is all nestlings. Yellow, we're showing the inbreeding coefficient for nestlings that had at least one immigrant parent. And in blue, I'm showing you the inbreeding coefficient for nestlings that had two resident parents. And you see evidence of inbreeding in the birds born to residents in 2009 through 2012. There's a decrease in inbreeding in 2013, and I'll get back to that. But for now, you can see that there's this really dramatic increase in inbreeding from 2008 to 2009. And what we think this is due to is this high mortality event that we observed in fall 2008. So in fall 2008, we observed the second highest mortality observed in our population over the past 30-some years. We sampled a bunch of these birds and found that 75% of the birds we caught had antibodies to Eastern equine encephalitis. The environmental conditions in this year were prime for high mosquito densities, and there were no observed increases in local predator densities and really high local encephalitis activity from sentinel chickens in Florida. And so we think that this high mortality event in 2008 was caused by some sort of disease, and this increase in inbreeding that we saw is some signature of an ephemeral bottleneck. But we do need to do more work to look into that. I mentioned that 2013 was weird because we saw this big drop in inbreeding levels. So if you go back and look at the immigration rate into the population, you see that in 2010 we had a really high number of immigrants in the population. These birds don't breed until they're 2 years old, and so in 2013 we had a bunch of birds that were born in 2010 breeding, and that might be why we see this drop. So I told you that inbreeding is increasing. Does this matter? We looked at hatching failure, so whether or not an egg hatches. This is one of the first real consequences of inbreeding in birds. We used generalized linear mixed models to look at a bunch of different factors. And the only predictor that mattered was the relatedness of the parents. So pairs that had at least one unhatched egg were more related to each other than pairs that had successfully hatched all their eggs. One thing to remind you is that Archibald has a seemingly healthy population of Florida scrub jays, and it's only through using genetics that we see any evidence of population decline. And so I've shown you that reduced immigration through time has led to increased levels of inbreeding, and this means that these small fragment populations may be more important than previously believed, and we shouldn't just focus on conserving these large, like a handful of large, healthy populations. This is just one of many ongoing projects, so I'm trying to look at kind of novel alleles being brought in by immigrants. We're looking at the effects of dispersal on patterns of isolation by distance. Stephanie Aguilon is a grad student working on this. She has a poster tonight. Go to her poster. We're looking for evidence of selection along the